this is a unique experience. I'm going before Joe Munzer for the first time. So normally MPS2 comes before MPS6, and I could rely on him to give all of the scientific background, the genetics background, and I could just fill in with some data. Today's talk is not going to be research based. Um, it's really a journey into memories and recognition, particularly of the patients and families that contributed to the MPS6 clinical trials. And um, the this is a unique opportunity to celebrate the 20th anniversary of Naglazyme approval and sort of honor both John Hopwood, who Steve alluded to, and I'll tell a little bit more about, and his developments in Australia, and BioMarin, who carried this project through beautifully, and then really the patients are the people who made this event happen. And it's still an amazing enzyme. I wish I could give it to every child 2 weeks after birth, and that's my hope. Someday we'll have newborn screening for this. So MPS6, it is, it's not quite as rare as MPS7, but it is rare. And so 1, 2, 3, 4A, they're all much more common. But it's unique in that it spares the central nervous system. So patients have normal development and are able to pursue normal lives. And so it was a uniquely useful enzyme to be able to treat their skeleton and pulmonary and cardiac and other manifestations that were really limiting their survival. So introducing two people, I don't know, is Tyler here in the audience? Here's Tyler sitting here. He has slowly progressing. We'll come back and say more about Tyler later, but slowly progressing MPS6 was part of this survey study. And then we have 3 of our patients that were in the phase 3 trial. You may not recognize at 12 years of age, Patricia standing to the right, and then our oldest patient, Brian, and Patricia's brother, Harold, who is 3 years older and much more severely affected than Patricia was at the time he came into the trials. So, looking at history and how we were able to get to do a trial for MPS6, we really have John Hopwood to thank. And he's not the only person worldwide. There were a number of groups working on MPS6, but he really drove the successful development of a CHO cell with the correct clone that could produce enzyme. He had previously studied the protein, characterized it, cloned it, and was able to then develop the production system. And then after producing the drug production system, he moved it into a CAP model. So these are people like David Brooks, Allison Crawley. He had a whole team of people that were helping him. Don Anson, who did the original cloning and production work. But you can see how important the large animal CAP model was in demonstrating that early treatment could really change the appearance. This is the face of a normal cat, and then this is an untreated cat with a sort of rounded forehead, short ears, and a treated cat with a much more normal appearing forehead. So they took this CAP model, were able to then move the enzyme in and prove that, and the enzyme could correct when started early, was effective. They looked at relationships of enzyme to GAG in tissue and GAG in urine. They were able to see the immune response to the enzyme and how to control it. All of this helped move it into human trials. They were able to identify the approximate dose that should be used. They had a feline aryl sulfatase B enzyme that moved into the cat and was 5 times as effective, and it took 5 times as much of the human enzyme, but it gave them an idea of how to scale it into human studies. So with John describes his 90s as a lost decade. He tried to move this enzyme into production with 2 different companies, both of which failed to be able to raise the money to produce it and develop clinical trials. So he lost, in 10 years, you almost lose a generation of patients that could benefit. And around late 1990s, the Emil Kakkis came to BioMarin with the MPS 1 Naglazyme, moved into trials, became a senior vice president at BioMarin, and during active trials, they decided strategically it would be really nice to be a 2-enzyme, not a single product in one area. And so they took a team to Australia. They were hoping to get MPS 2 clone from John Hopwood, who had already worked into partnership with TKT, so they settled for MPS 6. Thank goodness they were willing to take an alternative. And they licensed the clone and moved it back to California and began to develop a trial, develop production systems, which were really challenging. And there's a whole book that you can look at. Dan Marr has a book on the history of BioMarin, and you can read chapters about the struggles that they went through in these early biotech stages. Next slide, please. So give you a perspective. The 2 people, you know the top person well, that's Emil Kakkis, at least without a beard. Emil Kakkis, as we knew him maybe 5 years ago, 10 years ago. And then Stu Swedler was vice president of ClinOps. So he was running the trials. Emil was overseeing the whole program. And Stu was looking at different sites in the Bay Area and visited UCSF and California Pacific, a big private hospital in the city, and the CEO was favoring those institutions. Fortunately, Stu lived in Oakland. Nobody would ever imagine coming to Oakland voluntarily, but Stu lived in Oakland, so he was willing to entertain a trial at Oakland Children's. And we had some systems that helped us. We had more robust sleep study, and at that time they thought quantitative sleep study might be critical, but they wanted a trial in the Bay Area that they could oversee more closely than the partnership with Genzyme that was moving into a phase 3 setting. But you can see that from 2001, the start of phase 1, till they were moving into phase 3 by 2003. So each year there was a new trial starting, and each one had played a role in the decision process where we go next, or what the strategy was for the next trial. And between phase 1 and 2, they did a famous survey study that I'll show you a little bit. Call it the Swedler survey study. But okay, you have videos here, and I'll let the back play them, but we're looking at Mitch. Mitch may be in the audience also. Raise your hand. There's Mitch here. I've gotten permission to show these, and I apologize to the people we don't show videos from people I couldn't reach, or so it's a select group. But Mitch is doing his baseline on the left, and then probably one of our most severely affected patients, Kristen, is on the right, and these are the baseline. Kristen is 17 in this picture. I called Chet Whitley early when I was recruiting, said, do you have patients that might be interested? Would you be willing to contact them? And he said, I have two patients. One is Kristen, one is Kendra, and I'm not sure that they're in their late teens, and I'm not sure if they're still surviving, but fortunately both were still um, active, willing to come into trial. You can see Kristen, very limited walk, very limited stair climb. She actually had lost vision at age 2, which was pretty common. But you can get a perspective when you show the bottom 2 videos. Mitch is a little, as a 7-year-old, sort of intermediate. He's sort of on his, we activate the bottom 2. Kristen's now really walking comfortably. She's changed immensely with 6 months of enzyme therapy. And yes, this would be difficult to do a 6-minute walk test verification, but fortunately in this phase 1, we didn't have to do it. We did, it was a 6-person trial. There were 2 doses, the 1 milligram per kilo, 0.2. Both groups showed good urine gag response, although it was better in the 1 milligram per kilo. And it was difficult in that smaller group with the high variability to see walk test outcomes. But this just gives you a visual picture of a 7-year-old versus a 17, and probably more of an intermediate phenotype versus really severely affected. So here's our phase 1 team that participated. And you can see here's Mitch and his best friend Autumn, who's not here at the trial, Kristen, Kendra, and Julie Simon. I won't introduce this young lady. I was not able to reach her. And some of the thoughts that they sent back, Kristen's mother looks back. She may be here also. We were hoping she could make it, but Kristen was able to really get up and walk within a day after the first enzyme infusion. We had to report our first AE. Her joints were all popping for the first time, and so that was the initial AE of the whole trial. So it really was an amazing adventure to see her changes. And Kendra actually came into the phase 1, but told us she was having cervical compression and needed surgery, and they hadn't been able to put her to sleep. So we did not move her out of screening. She went home, and she came back for phase 2 after, after surgery. So, and then these same people, the fascinating thing is to see them 20 years, 25 years later, grown up. And here's Autumn. They were best friends in the trial. They were inseparable. Autumn is living in Portland, Mitchell in Minnesota, and they interact a lot. Mitchell has finished university. He was a computer science major. He's co-founded a company developing computer games, and Autumn works for the Oregon National Guard. She teleworks, and her co-workers are her cats and a dog. So, and then this year she wanted us to know that she got her first tattoo. So that was her big celebration. And this was a tremendous high point for me in being able to do a home visit. People don't think of physicians doing home visits anymore, but I had a chance to go from a genetics meeting in Nashville, drive to Knoxville, and visit with Kristen and her family. And you can see this is 10 years after the trial. She's in her almost 30 at this point, and truly remarkable, the extension of her life with this enzyme therapy. She's active. She's walking better than she did in the clinical trial. She's showing me how she types on her Braille typewriter. She communicates and counsels. Was a very religious person who did a lot of counseling and made, as Mom said, she made us all better. So it was a true gift to be able to visit in her house. She has passed away. She passed away at 39, about 2 weeks short of her 40th birthday, which would have been true. You know, people with longevity in this field, Joe Munzer would testify that this is almost unheard of with this disease severity. You see Stu Sweedler here. This famous Sweedler study was a cross-sectional of 120 patients with MPS 6 around the world. It got a number of sites that we knew would be phase 3 sites up and running. It identified most of the patients that came into the phase 3 study and obtained data a year ahead of when the trial actually started. But the key was looking at it, the biomarker. This is the urine gag. This is age in years, and it is one of the strongest pieces of evidence that urine gag is so predictive of disease outcome. Because when you're above 100, it's almost unheard of to have someone more than 20 years of age. So now we have many children, now adults in their 30s, and some that are actually reaching 40 at this point that were on the trial. So we similarly saw the same segregation. This is the rapidly progressing patients, intermediate, slowly progressing. This is height, female, male, and then this is pulmonary function, FVC volume. You can see the rapid and the slowly progressing patients, rapid, intermediate, slowly rapid, intermediate, slowly progressing patients. So a really wonderful biomarker, has to be done in a very standardized way, same lab, and before you actually start enzyme, you sort of lose the predictive value once you're in enzyme. So that led to a phase 2 study. I'm going to speed up. We had 10 patients, 5 in Oakland, 5 from Australia. This was the way John was not going to be part of the phase 3. They were worried about conflict of interest since he developed all of the treatment. And so we tried to recruit as many of his Australian patients as possible. Here you're seeing a group of patients in phase 2, 1 and 2, and then 15 years later at the National MPS Society meeting, same kids, grown up and enjoying each other's company. 2 of our phase 2 patients, Claudio lives in Lisbon. He's a graphic artist. He's still at university. He's learning Japanese. Severine actually has passed away, but really had a joy and improvement in life and activity and interaction with all of her siblings and friends. And then a special tribute to the group in Australia and to one of the lead participants in this trial, Bowen Oliver. You can see at 8 when he entered the trial, late teenage, early 20s when he's at the university and doing things like sailing at the Great Reef and walking the beach in Adelaide. So he actually passed away very unexpectedly during university, and it just shows we have a lot to still learn, and we need early starts, and we need improved monitoring, and it's really a difficult, comprehensive disease. And then phase 3 came out of the survey and what we learned in phase 2. You can see we went to a 12-minute walk test. It was quite onerous. There was a 92-meter difference in the change in the placebo versus the treated patients. And then in the second 6 months, the patients on placebo showed significant catch-up. And then the urine gag showed the classic rapid drop in the treated and no drop in the placebo, and then a drop after they went on treatment. So phase 3 went smoothly, except this sort of hides the fact that this was the one study where we got huge baseline differences, 100-meter difference in the baseline walk test, and it almost did the trial in, except a very bright statistician Janet Wittes was smart enough to build that into her statistical analysis, and it passed, was accepted based on that phase 3 trial. So we felt one of the high points of phase 3 was celebrating with John Hopwood in the center. He came and visited with our phase 3 patients. This is Anna. She's not a phase. She's the older sister of Isabel, who's shown here at the time she was in the trial, but she was actually in the hospital at that point. So she missed his lecture and description of the cats. And it was an international trial. The purple were all patients from that came to Oakland. The gold were international sites, and then Australia had the 5 patients. So Brazil with Roberto Giuliani, Portugal with Lisa Tellas, France, Germany, UK. So it was really a global effort to complete the phase 3, and still hard to find enough patients to fill. A little bit more about 2 of our phase 3. Brian was our only phase 3 patient that came from the actually came from the Bay Area. He lived in Napa, which is about an hour from Oakland, and he called with about 2 weeks left in recruiting. We were almost full for phase 3. He said, and he's an internet service provider, and he was on the internet with a friend in Norway who said, oh, you must be part of the Oakland trial. And he said no, and he started looking it up, and then he calls, and I found it hard to believe he had MPS 6. So I said, okay, we'll send samples to test. And he kept coming back. You've got to accept me. I've got MPS 6, and a little bit obsessive. And then finally, his last question just before he hung up, are there any women in the trial my age? And said, you realize that these kids have very normal teenage, young adult, all the same interests that all your other children have. And he was a force in the trial. He was almost 30 when he entered. He actually adopted and took care of our 2 patients from the Dominican Republic, Harold and Patricia, and their family as they adapted to U.S. Life and survival in the U.S. So these are 2 of my favorite adults, Patricia, who's on the MPS board, and Isabel, who is a star. They're both stars. Isabel, they both have graduated university. They both work. Isabel's testifying to Congress in favor of an immigration program for special medical needs. Patricia's graduating from university here and works in advocacy. So I will remember these people. Patricia and Isabel, they're both, these people keep me going. They're still angry. I don't come by as much on Friday. They picked a day when my nurse practitioner and I are both not around as much. So approval 2005 in the US, and then Europe in 2006. And we did resurvey 10 years later. Does it work over long term? And these were the walk tests on we had about half of the original survey patients. And this is the rapidly progressing group. And they're nearly stable over 10 years, and they slowly progressing are gaining over the 10 years. And a brief examination of survival, and it's difficult because you don't have many naive patients. This is the group with Naglazyme. This is naive patients that weren't treated, and there was a very significant difference in survival. At the end of this first trial, at the end of the phase 3 trial, we had the unique opportunity I didn't have, BioMarin, and Jim McGill had the unique opportunity to have an infant born just as the phase 3 ended, and he had an older sister with MPS 6, and Jim McGill organized with BioMarin to put both these kids on drug quickly. And so 3 years later, you can see the visual difference, same amount of treatment, but younger brother has been treated much earlier and has less of the MPS facial features. And then looking at their spine, we have severe scoliosis in the younger lady, a pretty straight spine in her younger brother. But even younger brother has cured the disease, and we still have a long way to go. And the question, and I've been able to help with a trial that Tippy McKinsey is ongoing at UCSF, where she treats with the known enzymes that we have approved in the U.S., including for MPS, in utero, probably around 18 to 24, 20, 30 weeks gestation for a start, and then every 2 weeks, and look in utero and at birth and for a year after to see if we see safety and feasibility. I think it's very difficult. It really, you have to identify the patients based on an older sibling that has one of the affected diseases. People have to still want to pursue the pregnancy, and it's relatively safe, but it literally will provide treatment significantly earlier. And we saw this on the first patient who had Pompe disease, and the family had had 2 previous children with Pompe disease, neither of which the muscle enzymes that are released at birth are lower than we see with newborn screening. The development has been normal. Patient still developed antibody. It didn't tolerize completely, but that was our time. I sort of looked at, I'm always asked, are people too mild to treat, too severe, too young, sometimes too old even? But I showed 3 of them, one because they were all in clinical trials. This is Tyler again. Tyler's 38 now. He had many of the problems that, even though he has slowly progressing type or phenotype, he has many of the complications, but they tended to occur later. This is Tyler from survey to resurvey in 10 years. You can see his height hasn't changed much, but his walk test improved dramatically on enzyme therapies. And other parameters improved with his pulmonary function as well, looking at absolute volumes. So without much growth, which is always why people challenge the pulmonary function studies, he showed a great change. And then Tyler now he's still a nuclear propulsion engineer, but most of his time is taken up with taking care of his 11-month-old and 3-year-old children. So he has a full life on Naglazyme. And who I've mentioned many times before, lives in South Dakota. She's well known. She's been through most of the wars with MPS and the different surgeries that are necessary. She had her decompression between phase 1 and phase 2, and she actually could sink a clinical trial because her improvement's so bad, so big, it would wipe out your statistics, your variability. But she went from 18 to 125-meter, 6-minute walk test. So it was a great response. Her stair climb improved, her flexion improved, everything improved with Kendra. So
I wish I had your energy.
This is Kendra giving a little bit of a description of her experience.
I should say, but the reality is being part of the trial was one of the best things.
Kendra, are you here?
Since I was 16, I didn't totally
Here's
know
Kendra over here.
what I was getting into. But looking back, uh, realizing what an amazing experience that was. I work partly with the med school, so I get to work with med students about rare disease. So I get to teach them about what I have, and they're always amazed
We
when
can
I
stop
say
the video.
part of clinical.
And Kendra has given a TED Talk, which I would never dream of doing. She's written books. She has designed clothing lines, and she has a full-time job working, developing programs for people with disabilities. So she's 40, which would have been unheard of before enzyme therapy. So then never too young, a unique story. Palestinian infant who was diagnosed at Hadassah Hospital, and we heard about it. We were able to have them. Dad and Amro traveled out when he was 8 months old. Dad was a construction foreman. There were 4 older siblings. Dad had never raised a child. Suddenly he had a 8-month-old infant on the airplane for a 13-hour flight, arrived without diapers, without formula. It was a real challenge. I had great worries he would not stick it out, but we were able to move him to the Oakland Mosque, had an apartment building that he was much more comfortable in, and he stuck it out. Amro was in the infant study, which studied 4 infants under a year of age, and now his entire family are 2 older brothers, older sister. Now he has a younger. Living in the U.S., have families, are naturalized citizens. So it's a great story.
Amro's 8 years old, and he
This is
started
10
coming
years
here
ago.
for weekly treatment when he was 9 months old.
I feel like good. And I feel like life.
Amro is hilarious. He's just like 40 years old in an 8-year-old body.
Dance makes me feel powerful.
I can't even describe it. It makes me cry. Dance Empower, what I see it does for these chronically ill kids is it's life-changing. I've been here for 32 years
we can
at Children's
stop
Hospital.
video.
I don't think I've ever.
And Amro is now in university. He's studying psychology. The whole family will mobilize and support him, whether he will be able to be independent like his siblings. We don't know yet, but it's a great story for a family from that would probably have not gotten treatment or maybe a bone marrow transplant in the Middle East. So still a patient. And then finishing up, this is earlier this year, I found out that some of our patients that we've just mentioned got together on a Zoom video to celebrate the 20th anniversary. So you see Isabel and Patricia, Autumn, Kendra, Mitch, and Claudia. So they're really happy. Final slide. In 2001, I got a message from 3 of our gentlemen in the phase 1 trial. I think they were all between 7 and 12, but they said, "Dear BioMarin, would you please make the medicine a pill? I wanted a pill so I won't have to get an IV every week." So we haven't succeeded with that. I think we've succeeded in every other facet, and maybe just around the corner there will be treatments that they don't need an IV. So finish by acknowledging again families, patients, their willingness to travel, to relocate, to disrupt their entire family to participate. John Hopwood, BioMarin, for supporting these studies, and providing this enzyme, and all the other investigators that I've been able to meet and work with around the world. Thank you. Thank you for listening. They can answer the questions better than me. That's we hope that the young generations are looking forward to the new trials and have a willingness to consider them, and they're always a challenge. They're
I had asked Dr. Harmatz if there was some point during the clinical study that there was such an obstacle you were worried you wouldn't be able to get past it, but then you were able to celebrate that you did. Is there something that stood out for you?
I think from probably my most nervous moment was the transition from phase 2 to phase 3. We were understaffed, and my lead coordinator, Julie Simon, who led the first 2 trials, was going to be too busy for phase 3. We hired someone. She was going on honeymoon for the first month that would be around enrollment period. Julie was going to cover a brownout hit New York. Her mother fell down the steps, broke her rib, and then ended up passing away in New York. And we were struggling to keep up with recruiting, data collection, and meeting regulatory requirements. And it taught me that you really have to have backup staffing. You have to have robust systems. It's sending patients back to sites, which we did a lot, Joe did a lot, and they're taking care of one patient, but all of the regulatory and all of the recording, and it's almost overwhelming to think of them doing a full-time clinical job and trying to be a clinical trial person on the side. I think you need a critical, it's not for faint of heart. You don't have the ability to put off an experiment for a week, or you're really rigidly on schedule and have to have people that'll be able to do it. So that was one moment. I mean, there were terrible. I can remember when I sent Kendra home, when we learned about the cervical cord compression just before Christmas, and I was driving off to Palm Desert for our yearly vacation, and Julie was left behind to pack them up. They brought everything out to be part of the phase 1 study. We're all settled in, and I had to take this incredibly good family and patient and tell them they couldn't come in the trial. They had to go back to Minnesota. They were motivated enough they were already had some of their care in Minnesota. They were smart enough to go and push them to correct the compression and come back again. But that was a terrible moment. We've had, like Oliver's sudden unexpected death. We had the same thing in a non-trial patient about a year ago, and unexpected, still unexplained, a very big star who suddenly had what looked like an asthma attack while they were swallowing a pill, and it was almost as if they were small airways swollen a little bit and unable to ventilate at that point. Taught me that anyone with a allergic diathesis should really have EpiPen available at home, whether they're having reactions or not. Just be prepared. So we never had a problem with infusion reactions to speak of. I have heard, as I've traveled, of almost, ones where you had to do real desensitization, and sometimes they would even give up because of the very long infusion times and slow infusions. So I've been very fortunate there. We learned a lot about, I think Isabel's, you have to go into surgeries well prepared and ready. You can't do it emergently. You don't want to do something in the ER or late at night when you have a different anesthesiologist on staff and you're trying to intubate and they're not your best person and you end up with a tracheostomy. So that, but we haven't had a CNS complication. We haven't had a surgical or anesthesia complication when it's been done with good planning. So those are some of my lessons. My wife helped me prepare for this talk. So I usually don't prepare because I don't like to go through and listen to myself practice, but she made me try, and I listened to her sort of frame what to say with each slide, and I can tell you she would have been much better than me. So it was a demoralizing
So,
practice. So
I-
thank you, everybody.
yeah, thank you, Dr. Harmatz.